physically be here in Seoul again. It's one of my favorite cities to visit in the world, and so yeah, happy to be here. All right, so I'm going to talk about some new work that, in the sense that it's just published, but actually some really old work in the lab. We've been thinking about this problem for quite a while, which is how to do actual statistical inference from single-cell RNA sequencing data. Here are my disclosures. So I think for this audience, hope all of you, especially with the talks preceding this, can appreciate that single-cell RNA sequencing throughput has continued to increase exponentially. And a lot of that has to do with experimental technology development and innovations. And some of it also has to do with these sort of like tricks that we've been able to come up with to try to get more cells into a particular experiment. And so this is sort of a set of methods that my lab published and also helped with in a few other papers, where we basically are trying to sort of, game the 10x system, where instead of capturing a single cell within a droplet, we're encapsulating doublets as well. And if you have samples that are labeled, in our case, samples from genetically unrelated individuals, then you can very reliably detect these doublets and then also increase the number of singlets that you get out per experiment. And so, these approaches have sort of like now been demonstrated over and over again. And there was a talk earlier from UC San Diego about utilizing multiplexing approaches to increase the throughput of your single-cell sequencing experiments. It also sort of, has other implications where we can generate better data. It's properly randomized for case control status or any covariates that you're interested in. And so, these approaches has really allowed us to scale single-cell sequencing even more. And, I think now it's fairly routine that in an individual lab, you can sequence hundreds of thousands to millions of cells per experiment. And so I sort of see this as a major opportunity for using single-cell sequencing as an approach to study various mechanisms. And I think most of us are still doing single-cell sequencing to kind of identify within a complex cell mixture the various different cells that's within that mixture. But increasingly, we can also try to induce variation by perturbing the cells with genetic perturbations using CRISPR-Cas9 or utilizing natural genetic variants across different individuals. And so here's, for example, a study that we completed a few years ago where we used genetic multiplexing to sequence about 1.2 million PBMCs from about 200 different donors. This is actually a comparison of patients with lupus, which is a systemic autoimmune disease, with some healthy controls. In total, we sequenced something about 5,000 cells per donor. So we showed in that paper that if you sort of do genetic multiplexing and analyze your data carefully, single-cell RNA sequencing can actually be very quantitative in several different respects. But the easiest way to sort of think about this is to look at cellular composition, right? So what is the frequency of the various different cell types in circulation? And, we actually compare this to existing, just complete blood count data, and it was very highly correlated, and which gave us a lot of confidence to make these estimates that in lupus patients of both East Asian and European ancestry, there is this lymphodepletion that's explained by the loss of naive CD4 T cells, and it's sort of a very robust signal. So that's changes in composition, but what about, changes in cell state? So given a particular cell type, is there a difference between individuals with a disease versus healthy individuals? And so I think this is where existing methods really are quite limited. And, my lab is as guilty as any other to try to sort of make sense of the data, often just by using exploratory analysis. So for example, when we first published this paper, we looked at the distribution of cells from patients, which is on the right, and then healthy controls. You can kind of see that within the monocytes, which are these blue population, it seems like the patient cells are in a slightly different region than the cells from healthy controls, right? So we can sort of claim that this is different. Maybe we can subcluster this and look for genes that are differentially expressed. But I sort of would argue that this is all kind of hacky, right? We're basically doing statistical inference by just looking at the data. But statistical inference is something that, of course, has been around for hundreds of years. And so the key sort of thing that we actually want to do is hypothesis testing, right? So, and what we really care about are questions around moments of distributions. And what do I mean by this? So I think most of us, when we think about any kind of sort of molecular quantification data, in the back of our mind, we're probably thinking about something like this, that you have a particular gene, and you have two population of cells, and that the gene is probably normally distributed, at least if you think about protein expression. And so you can sort of look at these two populations, and you can go, well, the average expression of these two populations are different. So that's the mean, right? So that's one of the moments of these distributions. But what's really cool with single-cell sequencing is you can estimate other moments. For example, you can have a gene that actually has no difference between these two populations in terms of the mean, but the spread is different, right? In this black population, there's more variability. And so we can quantify this with statistical variance. And when you do single-cell RNA sequencing, when you're measuring 30,000 parameters in a single experiment, you can also do pairwise correlations between genes. So gene 1 and gene 2 could be correlated in this group of cells in black, and then not correlated in this group of cells in gray. And in terms of the covariates, we could look at differences between cell types, which a lot of us are already doing, or case control status, which is the example I gave earlier. But perhaps even more difficult is when we start thinking about these genetic perturbations, either natural genetic differences between individuals or genetic perturbations that we introduce into cells. And these effects are often extremely small, where you can't just cluster data and identify groups of cells that separate out by genetic variation or induced genome engineering. So what's the problem, right? So if this is the reality on the left, what happens when you do single-cell RNA sequencing is that you're sampling very inefficiently transcripts within a cell. And so even if your transcripts are normally distributed, which one could argue that they're not, when you do reverse transcription within a cell, it's a hypergeometric sampling process. And in fact, even with the best estimates of sampling efficiency, which is anywhere between 5 to maybe 20%, a distribution on the left basically looks like a distribution on the right here. And so yeah, you can kind of tell that these two population cells have a different mean, but you can no longer, just visually looking at this, tell that there's a difference in the variance or correlation. And this is fairly representative of real single-cell RNA sequencing data, right? It's discrete. And it's even a bigger problem because we almost never sequence a library to saturation. We usually only sequence it to a certain amount that we can afford, and this is now a binomial sampling process. And so that's sort of like the fundamental problem. And so what do we do, right? And so what we do is if we want to compare differences in gene expression between populations of cells, we do pseudo-bulk because the data is so sparse. And so in this data set of lupus patients, we could do something like this, where we pretend as though we had done a bulk RNA sequencing experiment and can identify the differences between cases and controls. The cases are red and the controls are in blue. And this is if you aggregated all of the cell types together. But of course, with single-cell RNA sequencing, we can cluster first and look at individual cell types, which is great. And so we can identify, for example, that these classical and non-classical monocytes have a different set of genes that are differentially expressed compared to the rest of the cell populations. And so that's sort of like, taking pseudo-bulks of individual cell types that we identify in single-cell RNA sequencing, but we're leaving a lot on the table because ultimately we're not really using the entire data set, right? We're not using all of those replicate samples of monocytes within each individual as true biological replicates. And in fact, this paper that Square et al. Published now 3 years ago sort of really highlighted this problem, that if you looked at matched bulk RNA sequencing data with single-cell RNA sequencing data and ask how good do existing methods for differential expression analysis do in recapitulating sort of a differential expression result from bulk RNA sequencing of sorted cells, it turns out pseudo-bulk approaches where you pretend your single-cell RNA sequencing data is bulk performs better than all of these methods that's been developed for actually analyzing your single-cell RNA sequencing data. right? And so this is sort of a major problem. And so, you know, we had a number of ideas of how to possibly solve this, and really what made it all happen was an individual graduate student in my lab named Min-Cho Kim. He's a MD PhD student in my lab, but was trained in statistics and electrical engineering. And so he was extremely quantitative and thought about the problem and came up with sort of the following observation. And I think I can't click next anymore. Let's go here. Here we go. And so what he sort of observed is that single-cell RNA sequencing, actually he proved this, can be modeled as I mentioned earlier, a hypergeometric sampling process. And what that is that you can think about all the transcripts within a cell, and it's estimated anywhere from 200,000 to 250,000 per cell, and that when you're doing reverse transcription and having a probe to bind an mRNA molecule and convert that into cDNA, that's sampling without replacement, right? Once you've sampled that transcript, it's no longer there for other molecules to sample. And then the binomial sampling process of sequencing can also be modeled together with the sampling process of reverse transcription as a single hypergeometric parameter. And you know, once sort of you have that, what you can do is we don't make any assumptions about the distribution of transcripts within a cell, because some genes are highly expressed, like housekeeping genes, and they'll look normal. Other genes, like transcription factors, are very bursty, and they could look quite sparse. So we don't want to make any general sort of assumptions about distributions of genes in cells. Instead, we just want to model what we're actually doing at the molecular biology step, which is hypergeometric. All right, so this Y is basically the actual counts that we would observe, and then we sort of like normalize it by some quantity because of hypergeometric sampling, and that will essentially allow us to get estimates of the mean and then the variance and correlation between genes. Okay, so we basically take the observed data and recompute the estimates for the actual data. Okay, so that's great, but there's some problems with the hypergeometric. Most of these problems are related to the fact that we can't easily calculate likelihoods on hypergeometric or derive asymptotics with the sampling process. And so whenever you have something like this, you have to basically utilize an approach called bootstrapping or shuffling the data, right? And so why hasn't anybody done this? It's because whenever you think about shuffling data from single-cell RNA sequencing, especially when you get to hundreds of thousands to millions of cells, it can become extremely computationally inefficient. And so Min-Cho ended up figuring out how to do this extremely fast. So let me first talk about the estimators. And so the way that we really prove that this particular approach can work, and you know, from a purely statistical perspective, hypergeometric estimators can be biased, which means that the actual estimate could be a little bit farther away than the real estimate. And so we wanted to make sure that it's not so biased that we're not reproducing the ground truth. And what we did is we analyzed a data set published by Arjun Raj's lab, where they did both single-cell RNA sequencing with DropSeq, as well as small molecule FISH, right? So this is in situ hybridization of the same cells. And so what we can do is then use Memento, which is the method, and see how well are the Memento estimates for mean, variability, and gene correlation correlate with the same estimates on the FISH data. Okay? And so what you can see is that for all of these parameters, the Memento estimates perform much better than existing estimators, including for both variability and gene correlation, you know, a number of these imputation-based methods, which borrows information from neighboring cells. So just by modeling the actual sampling process, you can get better estimates compared to FISH without any imputation of the actual data. Okay, so those are the estimators. What about the shuffling? So the key insight in this, and this is something I think is still quite striking in my mind, is that, you know, it turns out just single-cell RNA sequencing data is extremely sparse. And so just how sparse is it? If you have, and this is a real PBMC data set, if you have something on the order of 30,000 cells and you have a matrix of 20,000 genes by 30,000 cells, the number of unique entries in that matrix that corresponds to a unique integer is on the order of 800, okay? So there's only 800 unique numbers in that matrix. But because of that observation, you can design a shuffling strategy based on the frequency of observing a certain count and not the actual columns of your matrix. And this basically approximate shuffling strategy will reproduce the actual shuffling, but at about 1,000th of the time that it would take. And so allow us to basically do very quick shuffling for a single gene. So for example, even if you have 100,000 cells, just to shuffle all the data for a single gene will take you about 3 seconds. And so actually 0.03 seconds. And so you can now imagine parallelizing this across a multi-threaded CPU to get shuffling times that are really, really fast. And so on average, if you're doing something like 50,000 or 100,000 cells, we can shuffle all of the data in 5 to 10 minutes on a single CPU. Okay, so what are the implications of sort of a better estimator and a ability to now calculate the confidence interval of that estimate? Well, the good news is that you no longer actually have to use pseudobulk anymore. And so here's Memento applied to the exact same data set that Square et al. Analyzed, and these two other approaches are also single cell-based differential expression analysis methods, and you can see that Memento is now first in class and beats out the pseudobulk methods on average. And it turns out some of these data sets have their own problems, and so we also analyzed an internal data set. Actually, it was the same as that lupus data set I showed earlier. What we generated is that for each of those samples that we did single RNA sequencing on, we also isolated 5 different cell types from the same cells and did bulk RNA sequencing on all of those cells as well across 200 people. And so, and you can see that in that data set, which is in many ways even cleaner, Memento does even better than the pseudobulk methods. Okay, so that's on differential mean analysis. What you can also do, though, with Memento is differential variance and differential correlation. So I just want to kind of walk through 4 different applications of Memento and show you guys some of the things that you can sort of use the method for. So this is a data set we generated during COVID, where we took some human bronchial epithelial cells that's grown on an air-liquid interface and stimulated them with both type 1, type 2, and type 3 cytokines, type 3 interferons, and did a longitudinal response, looking at the response of these cells at 0, 3, 6, 9, 24, and 40 hours after stimulation. And we're going to focus just on these ciliated cells since they're the most potent responders to type 1 interferons and also are implicated in COVID. So one of the things, the first thing you can do is you can use Memento and do differential mean analysis. And we can show, as we would expect, that alpha and beta interferons basically have very similar effect sizes on average expression of genes. And that's because both of them are type 1 interferons. And you can see that alpha and gamma are quite different because this is a type 2 interferon and this is a type 1 interferon. And then alpha and lambda are actually quite well correlated, but the type 3 interferon induces less of a response than a type 1 interferon. So this is all sort of known biology and just shows that you can use the method to do differential mean expression analysis, and we sort of already know this. What you can also do is estimate variance, and this is something that's really interesting that we sort of discovered in the data set. And what we're now showing is the change in variability between cases and control. So these are the changing variability in response to beta interferon versus the control cells. And so it turns out genes that reduce variability are some of the ones that are most overexpressed in response to the interferon on average, right? So this was kind of perplexing when we first saw this. In fact, this is an inverse correlation. But when you really think about it, it actually makes a lot of sense. So it turns out that some of these interferon responsive genes, when the cells are not being stimulated at all, are some of the most variable genes in the genome. But when you stimulate the cells with a cytokine, they all synchronize in their response. And this is in some ways how cells synchronize to each other, that we got a pathogen and we all should be producing this particular cytokine that neutralizes the pathogen, right? So this nice inverse correlation. You don't observe this nearly as much for gamma interferon, okay? So the other thing we can now do is differential correlation analysis. In fact, this actually further illustrates what I was saying earlier, is that these purple dots in the middle are all the genes that are highly variable across all of the cells, even at baseline. And so in response to perturbation, they become less variable, but these genes are already co-expressed even in cells that are not stimulated with any cytokines. But in response to cytokine, as you can see that this changes dynamically over time, especially at 6 hours and 9 hours, it seems like there's a bunch of other genes that are now co-expressed with that core module of interferon-stimulated genes over time, and the co-expression goes away by 48 hours, right? So this is now rewiring of a gene regulatory network dynamically over time. And you can sort of, identify these relationships only by doing differential correlation analysis. So the second application is a data set that we generated internally where we sequenced 170,000 CD4 T cells. But what's unique about these cells is that they've also been perturbed. So these cells are edited at 280 locations in the genome, corresponding to about 140 transcription factors. And we did single-cell RNA sequencing all of this. So this is done with CropSeq. And these 140 transcription factors are chosen because they either are highly expressed in response to activation in CD4 T cells, or because the regulatory elements that these transcription factors are thought to be binding to are highly active in CD4 T cells, as estimated by bulk TAC-seq. And so you can see that on the right, overall, the experiment worked. And this is Gen 1 of the CRISPR technologies in T cells, so it's surprising that it worked as well as it did. But there's this nice blue diagonal, right? So cells that are cells where we have a particular guide RNA expressed, if you look at the particular target gene, those cells will express lower levels of that target gene. So this is nice positive control that the experiment worked. But here's the kind of analysis you can do. So if you just do differential mean analysis, you can get something on the left, where basically each column is an average expression profile for each cell that has a particular guide RNA in it, and then each row is the average expression of all of those cells. And so you can see that, there's some of these really interesting regions that kind of light up. And so, for example, this cytoskeleton proliferation genes are turned on when these particular guide RNAs are introduced in cells, but so are these autoimmune Th1 and Th2 differentiation genes. And so you really can't tell that there's a difference between these two modules of genes if you just look at the average changing expression in response to perturbation. But now, if you look at the gene-by-gene correlation matrix, and this is a gene-by-gene correlation matrix made in cells that were not perturbed, so these are wild-type cells, you can see that it's actually quite different, right? These two modules of genes. These genes up here, the Th1, Th2 differentiation genes, are correlated with each other, and then this module of cytoskeleton proliferation genes are correlated with each other strongly. There's some cross-correlation between genes, but you can see that the clustering informs us of the differences between these gene modules. And so adding this additional layer of correlation can help us further interpret and map gene regulatory networks in response to perturbation. Something else we can do is now actually combine the differential mean and differential correlation analysis to detect epistatic interactions. So if you just look at the differential expression analysis results, basically what we can generate is a map like this, where each perturbation on a particular regulator, either R1 or R2, if it changes expression of the gene on average, we can sort of draw a line, an edge between the regulator and the target gene, okay? And so this is sort of a nice network, but it doesn't really tell you anything about causality or potential interactions between regulatory elements. But you can also do something like this, right? Because you have single RNA sequencing data on the entire transcriptome, what we can basically ask is, if we knock out regulator 1, does it change the correlation of regulator 2 with the target gene? If the answer is yes, then we can hypothesize that these two regulators may interact with each other. If the answer is no, then these two regulators potentially interact independently to regulate this particular gene. And so we can identify these candidate epistatic interactions, and we were able to validate some of them. So the third application is, we'll go back to that lupus data set that we generated. This is a data set that was collected on 200-some individuals, and each of these individuals were genotyped. So now we can look at the genetic control of these various gene expression distribution parameters. So the first thing, of course, is we can look at its effect on mean differences in gene expression. And what we were able to show is that this is basically mapping basic EQTLs in each cell type, is that Memento has increased power compared to even a pseudobulk-based approach. But there are things that you just can't do with pseudobulk, which is to map QTLs that control variability, as well as QTLs that control co-expression. And I'll show 2 examples of this. But let me just first talk about the mean QTL result. So what does this power increase allow us to do? Well, as sort of Tim mentioned earlier, one of the things that we were really excited about when we map QTLs is that the hypothesis is that if there's a genetic variant that's associated with gene expression, right, we expect that this particular variant could be modulating or perturbing a chromatin accessibility region in a particular cell type. And so if you do the pseudobulk analysis, you can see that there is this diagonal where EQTLs that are active in B cells are enriched for regions of open chromatin only detected in B cells. And that's true for T cells as well as CD8 and NK cells, which are pretty similar. But now if you look at the Memento results, this enrichment is much, much stronger, right? That there's much more cell type specificity for QTLs that we detect in B cells enriched for regions of open chromatin. What we can also do is, basically look at the rank sum statistics of explaining particular GWAS associations. And again, Memento produces much higher enrichment in each of these cell types compared to pseudobulk. And so I'll just give you 2 examples of some new sort of quantitative traits that we're still trying to figure out what they really mean. For example, here's a genetic variant that's associated with not just the average expression of HLA, but the spread of expression. So you can see that individuals who are homozygous for the A allele have much wider distribution for this particular gene compared to individuals with the G allele and heterozygous individuals. So this could potentially hint at epistatic interactions between this particular variant and some other gene. We can also identify co-expression QTLs. So this particular variant that's actually located in a cis-regulatory element of this gene LYZ controls the correlation between this transcription factor GUNB and the expression of LYZ. Okay, so individuals with the G allele have no correlation. Individuals with the A allele have great correlation. This particular case was easy to explain because that cis-regulatory, that SNP, that single nucleotide polymorphism, is located in a GUNB binding site, right? And so we have lots of examples of this. And so, we can't always explain it as a transcription factor binding a cis-regulatory element where there's a SNP in it. But this is sort of a nice explanation for why there's no correlation. And so as a last example, what we can also do with Memento is that shuffling approach that I described can be sped up even more. And so what we can basically do is go to a census, like the cell by gene discover census, which currently has about 100 million cells in it, and we can sort of regularly, maybe every couple months, shuffle all of the data, right? We don't want to do this every single time we want to do an analysis. We'll just want to generate a null distribution whenever there's sort of a large update to the database. And when we have that null distribution, then we can power essentially arbitrary comparisons across multiple data sets. So this is actually our data in the cell by gene corpus, the lupus data set. And so you can, compare the different expression differences between CD4 and B cells, but you might also want to make comparisons between a lot of other cell types. And so you don't want to recompute your null statistic every time you do this operation. And so by pre-computing, this can be made much faster. It took a lot of engineering and in collaboration with the cell by gene team, but we show that a pre-computed p-value is very well correlated with the actual default p-value computed with Memento, which is already very fast. So this default is already using that fast approximate frequency-based shuffling. But this pre-computed one is even faster. And this is true for both mean and variability. And in fact, it's so fast to do the pre-compute that we can't even really, it's just essentially instantaneous. And so what that will allow us to eventually do is that you can go to a website and get near instantaneous report back of differential expression results. And so we test this out a little bit. So, why would this be useful? For example, we wanted to do a comparison between what are the genes that are differentially expressed between plasmacytoid dendritic cells and conventional dendritic cells? One of the reasons we want to do this is because PDCs in each data set is about 2%, right? So they're actually quite rare. And if you can combine across all the data sets where you sample this particular cell type, then you can improve your power. And so this is what we are able to show, is that the joint differential expression analysis with pre-computed Memento has better power, and that if you use each of these individual data sets, right, you'll have less power. Okay, so I'll just end with sort of like this video of what we hope to release. So a lot of the back end of this is already available. So many of you might have already played with this. You can basically go to the cell by gene, discover, pick up any arbitrary data set. So for example, this is our data set again. And you can choose sort of a random set of cell types that you want to compare. Actually, we're doing a case control comparison right now in a particular cell type. So you can take CD4 T cells, compare the expression of CD4 T cells between lupus patients and healthy controls. And it's fairly instantaneous right now, where you can get a result back. It's reporting out the T statistic results, which is computed with asymptotics, but we're working on the cell by gene team to also report this back out for Memento in the coming months. So we're pretty excited about delivering this capability to the community. All right, so I'll just end with the summary and sort of a few thoughts. So Memento is a new method for hypothesis testing for differences in mean, variability, and correlation from single RNA sequencing data. Hope I've been able to convince you that it's accurate, it's robust, it's also super efficient, scalable. The details are described in the paper. We show an application of Memento to look at the response to stimulation, perturb-seq, and QTL mapping. You know, and I think it's important to sort of like emphasize that statistical inference remains important and that methods matter. So I think there's a lot of exploratory analysis of single RNA sequencing data, and there's also lots of prediction based on single RNA sequencing data. But statistical inference remains, I think, one of the core sort of operations you want to do as a statistician to ultimately understand mechanism. And so let me just end with sort of this example, because I gave this talk actually at the NIH, and the person who spoke before me was the person who won the Nobel Prize for the Hubble telescope. And you know, and hearing the talk, it sort of reminded me of this, right? Like, you know, when you look through a telescope and you see this, which kind of looks like a UMAP, by the way, you know, how many stars are there, right? So if you just do inference using sort of visualization, you'll go, well, that looks like 5 clusters. I don't know what the mechanism is, but it's got to be 5 different cell types. But it turns out that there's only 2 cells, and this is, or 2 stars, and this is an example of gravitational lensing, right? So the mechanism can produce a result that is not what we actually expect. And so, and by the way, I think it's also important to sort of take a look at this paper, which came out of Michael Jordan's lab, is that you can actually do inference with prediction methods, like deep learning methods as well, with some significant limitations, and they do a really good job of describing what those limitations are. All right, so with that, happy to take questions and acknowledge Mincho again, who single-handedly did all the work, and he's now in the ER being a real doctor. Thanks.
All right, thank you very much. So please feel free to ask questions. If you have any question from the floor, please use the microphone in the middle of the hall, please. And also use the chat room.
Hi, Jimmy. Thank you very much for a super interesting talk. And my question is about the cell type part. Like, is this Memento replacing the HR and DSEQ part in Suderberg differential expression testing, or do you kind of do the soft assignment of the cell type in this statistical inference? That's my
So
question.
I think you can use it in both ways. You know, if you're really sure about your cell types, sure, it can definitely replace the HR part. But I think it's really interesting that if you don't, let's say that you just take PBMCs, it's from different people, and there's a compositional difference, it'll probably look like a difference in variability, right, between individual samples. So I think it could be used to identify essentially like heterogeneity in the data as well, in a statistically principled way. So then you follow up and figure out what the mechanism is. But you can use it in many different ways, and very sort of like, you can make some predictions about what you expect to see.
But then have you thought about making it automatic? Like if you start from the entire bunch, then you'll be able to see what kind of cells are showing the genes are showing variability changes, and then maybe you can zoom into those and see which portion of the UMAP they are showing the like mean differences.
Yeah. So, I mean, this is where like statistical inference does have limitations, right? I mean, I think like, you know, it's like even with bulk RNA sequencing analysis, it doesn't tell you the mechanism. So we can't really automate that, but we can give you very strong statistical guarantees that this is what you should be following up on.
I see. Thank you very much.
Okay, we have question from the chat room. So the question is regarding handling sparse data with Memento. So could you elaborate more on how the method manages analysis with low memory usage, please?
Yeah, so the idea is that, you know, so if you think about the number of unique counts in a data set, given the number of cells you have, right? If it's logarithmic, basically we have a shuffling strategy that is memory usage on the order of, it's like logarithmic the number of cells. Because you don't need to shuffle each column, you just have to shuffle the frequency of observing a particular count in your data, assuming that all the counts or the frequency of observing a particular count in a particular gene are independent from each other. And that's generally true because we're just modeling the sampling process. I think that's the right answer, but the details you should get from the paper.
Okay. So any more question? Oh, so we have another question. So how do you feel about deriving cell type composition insights from solid tissue sample single cell data? For PBMC data, I can wrap my head around how differences in cellular composition could represent biologically significant variation, as I would assume circulating blood to be a form of relatively homogeneous solution. However, solid tissue isolates may really depend on which part of the organ is sampled unless the whole organ is dissociated. Can having enough samples compensate for this variability?
I mean, you know, this is sort of like the classic quote, right? Like you can't ask a statistician to perform postmortem on a bad experiment. I mean, like if you're not sampling correctly, it's going to be pretty difficult to make sense of it. Maybe if you collect enough randomized data, you can do that. But it seems like some of these in situ methods are going to be pretty useful. So you want to minimize whatever is introducing that bias. We think the model for Memento can also be used for spatial analysis because you have the same problem where you have very inefficient sampling of transcripts. Even I think for Xenium, the estimate is like between 5 to 10%, right? So the same sort of statistical framework can probably be used to identify spatially differentially expressed genes. And so if you have lots of spatial maps, I think that might be a better way to deal with complex tissue.
Okay, so if you don't have any more questions, we will
Thank you.
Wrap up the talk. Please give big hand for the great talk to Professor Jimmy A. So the last speaker of today's symposium is Dr. Fabian Theis. Professor Fabian Theis received PhD degrees in physics and computer science in 2002 and 2003, respectively. After working as a postdoc in Regensburg, Tokyo, and Tallahassee, he took up a position as Bernstein Fellow at the Max Planck Institute for Dynamics and Self-Organization in Göttingen. He then joined the German Research Center for Environmental Health, Helmholtz Zentrum München, where he was a group leader at the Institute for Bioinformatics and Systems Biology for 6 years. In 2009, he became an associate professor at the chair of applied mathematics at Technology University of Munich. Since 2013, he has been a full professor of biomathematics at TUM, where he holds the chair of mathematical modeling of biological systems and director of the Institute of Computational Biology at the Helmholtz Gentrum Mansion. He will give us the lecture on the topic of generative AI for learning cellular state and response from single-cell genomics. Please welcome Professor Fabian Theis.
Hi. Yeah, great to see you. Sorry for not being able to come in person. We have a little vacation here at the moment, and kids are around. I hope they're not coming in now, but I'm very excited to be able to talk to you this morning. Thanks for interest in that. So I want to talk a little bit about how we use generative AI mostly to model cellular state, a little bit about response, but then also sort of to get a little bit interpretation in. And I think this idea, you know, this revolution, I mean, I think in our field in computation, I think generally in computation biology, we've been using machine learning for a long time, right? But, you know, being generative about it, I think it's sort of interesting. Kind of ties a little bit into excitingly what Jimmy was showing before, that you sort of want to add statistical inference, really get sort of maybe variational error bounds, things like that to whatever you do. And this is old saying from Feynman, famous physicist. They said, like, what I cannot create, I cannot or do not understand. You know, this sort of, you have this forward physics-based models where you understand things. And you could argue that if you generate something, you do create, but, you know, people would maybe more call this black box. So I think it's really interesting back and forth between these two aspects. And I think maybe for our purposes, often it could be a good model of a cell that sort of, if it behaves well under perturbations, you have a prediction. Is that maybe where you want to go? Or do you want to have sort of really detailed pathway mapping? I think both sides can have an argument for it. I think it's interesting where this will hold. This is from an older review we did with Nicholas Rejewski, this picture of cells going through this high-dimensional state space, going through various decisions if they sort of take a wrong turn. I think is really exciting. I think a big part of our research in single-cell biology, I think, would be to learn these cellular landscapes and then be able to act upon it. That's kind of what you could call generating, right? So you want to sort of see what happens when your cells maybe sort of make this trajectory, then you can use trajectory inference for that. Maybe what happens if, you know, sort of cell takes a wrong turn, goes into one particular disease state, you know, can you maybe then sort of understand this landscape part, and can you maybe act by sort of finding the ideal perturbation to go back? And, you know, these perturbation scales, they're actually, you know, these assays that allow you to do high throughput perturbations also on single-cell RNA-seq level, which is sort of what we mostly focus on, I think might enable us to address some of these questions now. Before I start, I just want to sort of really briefly say that engineering matters. My lab has been developing ScanPy for analysis, single-cell analysis in Python. And because, you know, to really maintain a growing ecosystem, we realized we need to really partner up with colleagues and friends, where we set up this SC-Verse consortium, which now has a whole bunch of shared data structure, but a whole bunch of core packages. And then basically the Python single-cell ecosystem around that sort of interacts with these packages. And we invite you to contribute or work with us if you're interested in this. We recently had the first SC-Verse conference, really very nice growing community, very international. And I think it's sort of exciting directions coming in there. For example, now scaling everything up, our 2.0 release next spring, summer will be to sort of run ScanPy on at least 1 billion cells. I think these scales are coming up, but then you get everything GPU accelerated. So these engineering aspects are important. We've been doing things for spatial analysis, particularly also a multimodal spatial framework together with Olli Stiegler's lab. I think important sort of baseline, sorry for that, baseline data structures to then build upon with potential machine learning models. We've been recently releasing PerPy, a perturbation package where you can sort of also visualize then perturbation effects in sort of a one level up sort of in perturbation space, which you can sort of see as a population representation. And also been talking about how to do these sort of how to do analysis. So this is just part on the engineering aspect, which, you know, so many of us I think are dealing with to get things useful. But I want to talk about this action on these cell landscapes. And with my intro, you might have seen already that, you know, a beautiful review from Croatia and Peter Sorgo some time ago. They called this differential biology. But, you know, there's this sort of this back and forth between these principle-based models that we, in biology, because of all our priors, often take, and then the data-driven ones. And this may be sort of the hybrid way to go, are there back and forth. And I want to share with you that we can maybe take some data-driven models and make them a bit more principle-based or at least interpretable. The type of framework that we use, of course, is going to be deep learning. I think with the scales that we see, I think is somewhat inescapable. These type of autoencoders in our field that sort of take these gene times cell matrices, then learn them in an unsupervised fashion, were first introduced by Yusuf and my lab sort of in parallel, where essentially sort of minimize the reconstruction loss, and then you sort of get this sort of cleanup data in particular, but you get this bottleneck layer that's sort of interpretable. You can add conditional autoencoders where you then sort of give that information, thereby sort of removing, sort of being able for this network to use this information, thereby sort of in this bottleneck, removing this batch effect, sort of have this most efficient encoding. And that's a very useful technique for batch correction, data integration, and so on. It gives you then these transitions, for example. And here's an example of really just a two-dimensional bottleneck for PBMC data, where you see that they really pretty much look like a t-SNE plot, so or UMAP plot. So they really identify the core structure in the data. I think it's very exciting. So then, you want to interpret these latent spaces. I think what's important is that these bottlenecks, though, like each dimension in itself doesn't mean much. They just sort of encode the data. But by adding priors, sort of, going from this purely database one to maybe a hybrid one, you can start interpreting it, adding perturbation effects, things like that on. And if you want, in a very weak fashion, just even telling it about batch effects is sort of some prior knowledge, right? We, together with a bunch of colleagues, have been running this data integration competition at NeurIPS, and we've been actually doing this now up to now, also with perturbations and so on. And we see that these things are quickly taken up sort of leading ranks just because they're so flexible. And I recently read from Pass Bio, I think they've been saying that nowadays a single paper has more than maybe 500,000 cells or so in as sort of a new contribution. So these scales are also getting big. So I think it's going to be useful to take up these flexible models. But where's this generative? Do you guys remember this picture of the Pope in the funny jacket? I've been showing this a while, but this is an example of generative AI, which is not actually really it's not real, right? But how has it been generated? Well, because there's real pictures of men with white coats, there's real pictures of men, and if you sort of take the difference of these, you get this white coat, you add them, so you generate them. This is sort of how these generative picture models work. How can we use these generative effects in our field? Well, this was the idea that we've been sort of proposing some time ago, where you take now a cell under a perturbation that could be sort of maybe something that you measure, right? And then you subtract the ground state, and then you sort of have this new set of cell lines where you want to sort of extrapolate where this effect would go. And this is then how you can also be generative about it. So you can sort of do this latent space arithmetics visualization that I showed you before. Also in single-cell RNA-seq space, we initially proposed that it works to some extent, but of course, you sort of need to do more to get this to work. How is that principled idea? I wanted to talk about perturbation effects. I'll be mostly speaking about this gene expression space in a minute, but I just wanted to review this part briefly. If you see transitions of cells in a few examples, let's say this could be different cell types, what we do is then, for a new one, if you want to predict that perturbation behavior, just as I showed you with this latent space arithmetics, you encode them, you go this latent space. In this latent space, then you expect that these things turn linear. That's kind of what we did with this addition effect. And if you then have a new one, you apply this perturbation and you go back. Of course, there's a big assumption, right? Why would they be linear? Computer vision, in some examples, it was shown that this actually makes sense. Often nowadays, we want to actually put additional priors in to make that happen, right? And of course, then we can sort of predict this. So in a few extensions, in particular also one with Stefan Günnemann and one with Meta AI, we've been extending this to add sort of embeddings of these deltas. So this would be, for example, a drug space or also CRISPR space, but then also making sure that you get these linear transitions. And I think the most recent one where we work with optimal transport type of constraints is a very exciting way to go. And we're recently combining this with diffusion-based models that are sort of the most expressive versions in computer vision to really make this happen. I won't be showing these results, so this is basically the ongoing stuff, but I think this is already useful. But what you will learn in many of these examples is that they generate and sort of behave well for letting sort of supporting maybe an experimental design, take it the next step. But this out of distribution generalization, this is sort of the key task that you want. You want to sort of be able to go into a new, let's say, new patient, new direction that you haven't been measuring, that's hard and really needs massive data. And there could be an argument that we might not be there yet in single cell perturbations. And already in that early paper, we found sort of linear methods to be performing well in CPA. We benchmarked this as well. And of course, you also don't model the dynamics, right? Remember, we had this Waddington's landscape idea, like how do you see that here? So I think there's room for extension. Just to summarize this part briefly, if you want to model a cell's response to a drug, right, you can use these generative AI techniques to extrapolate this response profile. But we have all this information. Central dogma could be one, right? Like, how can we add that information? And for me, one of the most beautiful examples how that works, sort of these really prior systems biology type of approaches in our field is RNA velocity, as has been initially introduced by Stelinissen and Peter Kachenko a few years ago. This idea being that you have this splicing metabolism that you now measure, and you sort of assume it's independent in that simple system across all genes, and then you just write up a very basic differential equation. But because it's decoupled, you can actually solve this in closed form. And then if you sort of plot unspliced versus spliced, you sort of get this dynamical behavior, right? So can you combine this with these latent space techniques, right? I mean, because with this one, you sort of estimate rates, you embed them, and then you sort of get this sort of vector field, right? Very, very useful thing. And recently with Nir Yosef, we've been actually combining this sort of estimation technique here with a way how to put this into a sort of combined perturbation space where you can then sort of see where these transitions not just embed, but where you can sort of also get an uncertainty model for that. Still, this assumption that these things are decoupled are there. So it's not, so it gives you sort of this dynamic behavior, but it sort of doesn't give you the wiring under Waddington's landscape, as you might have seen. These are like an underlying GRN or things that push that direction. In parallel, there have been a whole bunch of techniques that deal with the idea of learning regulatory networks instead. So not just doing this, but actually sort of seeing where a perturbation would go. There's, for example, Cell Oracle from Sam's place. I think she was talking earlier. Dynamo from John Weissman, Pando from Barbara Treutlands. There, we've been doing cell rank sort of interpretation of these dynamical models. But these things work well in terms of sort of getting the wiring in, often from single cell attack or also databases. But the true dynamical nature is not really interpreted. So you don't get the dynamics. So you have this RNA velocity, you have this regulatory network, so can you sort of combine? This is sort of bit the idea what I want to show you in as sort of one of the first things we've been doing. So here, what you can do is you can write out the dynamics of sort of the full transcriptome state in a cell as of a function of its previous state, right? And that could be, of course, just gene expression, and then this F somehow needs to encode the dynamics. This is, of course, challenging. That's why people usually don't do it, because like fully estimating that means estimating a 10,000 dimensional space across like hundreds of thousands or millions of samples nowadays. So that's why we usually assume this thing is decoupled, right? But we think that we can put these things now together if we assume a regulatory prior, and we call this RegVelo. Let me show you briefly the idea how this works. This is, by the way, work of really talented PhD student Wei-Shu Wang, who has been working on this for last 2 years. So what this does is it bases itself on that embedding model that you saw before from this collaboration with Nir. So, but in the same time, we also assume a prior regulatory graph, right? So this prior graph we then use to constrain it. For these unspliced splice matches, we do the typical encoding decoding. We get the latent state, and then from the prior graph, we know basically for each transcript, based on these edges that we sort of take from the graph, a particular level of transcription rate. This transcription rate level we then put into the dynamics, in the dynamic model that you saw before, but now we couple it. And we actually also have a sort of soft version that sort of allows us to extend this graph beyond what we just originally have. So altogether, we get this regulatory kinetics, and then we can write down a likelihood for a particular set of observations and then fit the data. The key problem is, of course, the parameter inference. And in this case, we just use variational inference, but because of these sort of high dimensions, sort of the key contribution, I think, would say that instead of now fitting 10,000 one-dimensional ODEs, which actually you have a closed form, you now have this coupled one, so you need to solve this on the fly. And the trick is that you have these very flexible neural network type of inference models that can basically reduce this to neural ODE and then solve this. So this was really the major achievement, I think. So let me show you what this thing can actually do. So for example, here you now see a single cell a model where we actually have a label for cell cycle just with a Fuji marker, right? And we can now see how the dynamics of rig value look like, and they kind of seem to be sort of correlating nice with the marker, but we want to be quantitative about it because we want to sort of compare with the non-regulatory network prior based models. So for this, we measure things such as cross-boundary correctness, where you sort of go through the cell cycle phases, some confidence in the velocities estimated in correlation with the known prior. And then we see that we get sort of robust values here. We can compare them in a minute to other methods. Similarly, because we don't only estimate dynamics, but we also estimate a regulatory network, sort of adapt the given one, right? We can ask how well that sort of fits to known sort of states in that cell cycle dynamics, and we can sort of look at the edge predictions and particular weight prediction models. And if you now compare this to existing models, just for the velocity side first, you see that this rate value approach outperforms with respect to a whole bunch of scores this inference, consistency of velocities, and also the latent time. And the edges you sort of now can compare to models such as Cell Oracle or what we showed before. Using the dynamics essentially helps it to sort of get also better edges. So it's sort of this constraint based on regulation, but allowing it to smoothly extend it allows it to build both better networks, but also because of that constraint, inference more dynamic, more robust dynamics. Really excited about that. We can do this now. The beauty now, of course, is if you have an underlying sort of more prior-based model, you could be much more robust in going out of distribution. Remember before I said in these purely data-based ones, based on, let's say, diffusion models or so, we need a massive amount of data to be OD. But here now we have a prior, so we can just basically take a network and sort of mask some type of regulon, right? And then simulate the dynamics, look at the local effects, and then we can see how the original dynamic looks versus the perturbed one. So we just look at the perturbation versus the original one. And then you can sort of look at the effect of particular perturbations. So let's try this. Here we do this for an example from pancreatic endocrinogenesis, where you have here progenitor cells that then go through sort of early pancreas development and then form different cell types in the islet, such as beta cells, alpha, and then also delta and epsilon cells. And by just sort of looking at the dynamics of the vector field that rate value estimates, but then applying a cell rank on top of it, you get the terminal states as you would expect them. So that works very well. And we can actually also show that this sort of number of terminal states that we get sort of works much more robust than compared to sort of our previous approaches of VeloVia and SCVelo. But in addition, you can now do knockout simulations. So you can take this one and you sort of do a knockout, for example, of NeuroD2, and then you see sort of that the epsilon cells don't exist anymore, as is known before. And then you can sort of look into that screening of this and can say, what type of targets are actually mattering, and you sort of look at these particular ones, and can you do, for example, type of double knockout, and you actually see that just sort of most of them get alpha cells. And these things can be confirmed from literature. So that's, I think, quite useful for us. Let's now really look into a data set where you have sort of measured perturbations. In this case, we're really lucky to pair up with Tatiana and Siwan from Oxford, who have been looking into one system for quite some time, namely the zebrafish neural crest development, where they have a time course, both SmartSeq2, so very highly quality resolved full transcripts, as well as 10x multi-ohm cross time points of this developmental system. In addition, they've been able to set up also a transcription factor perturbation setup where they can directly capture then in a droplet-based model these knockouts. So it's essentially like an in vivo perturb-seq. So first we can learn latent time. So you see UAP, I don't show you the annotation, but because we have different stages, we can sort of see that this learned latent time from Rigvelo nicely associates with the staging. So the latent time sort of gets grow up with this, and it's typically expected that they often grow wider, that there's some cells that will stay more early here. You can do terminal state identification, and then you get sort of hit missing pigment cells and so on as neural crest cells sort of then need to go in development. And you can show that you actually identify these sort of terminal states much more robustly than with previous approaches. But now you can do the perturbation, right? So you have a pseudotime, you have a model of regulatory dynamics, but now you can do perturbation. For example, here we look into a few driver genes for mesenchyme, and then we see that if we do various knockouts, you actually get sort of an enrichment of particular cells and depletion of, for example, hit mesenchyme, as would know if you kill those mesenchyme driver. And you can do the same thing for pigment cell drivers, and you sort of see a knockdown in the pigment ones. So you sort of get the right knockdowns as you would expect from literature. But because now we have this perturb-seq, right, we can actually also really look into the whole set of new factors that Tatjana was just hypothesizing. And what we see there is that in such a perturbation benchmark, we, for example, find that our rake velo, and there's sort of two versions of it. We have a soft one that sort of allows you to learn additional edges, and a hard threshold one really gives you much nicer correlation with the observed perturbations than dynamos or cell oracle or something like that. And this is consistent with a single knockdown as well a combination knockdown. So just to summarize this, with this rake velo, we kind of have this hybrid model that takes prior information from a regulatory setting, like from a gene regulatory network or something like that. And typically, as we say, we use, I think now mostly scenic plus for inference from either given attack or previous one, and from a dynamic one by combining with velocities. Of course, there's a bunch of limitations still. The underlying regulatory dynamics are still quite simplified. So we have an additive sigmoidal model for that. So there's, I think, a whole opportunity for including epigenetic modifications. And I think adding additional modalities, of course, is going to be challenging. So to summarize this, how to model a cell's perturbation dynamics, I think it's really useful to write up a forward model, and with these powerful frameworks, we can actually also now do inference on this. So we combine this deep cell representation with these gene regulatory network. The second, and this is unpublished, so we hope to bring this out in the next few months. The second unpublished thing that I wanted to share is a representation learning. So yeah, obviously we want to learn atlases, right? And we've been doing this in human cell atlas. And one sort of example that I want to show you in a minute, where I think we can find better representations, work by Lisa and Malte in a big collaboration with the whole Human Lung Cell Atlas network, which is the Human Lung Cell Atlas. We've initially taken a bunch of data sets, harmonized annotation and metadata, automated location, and build these core atlas that with transfer learning, then we extended to, I think, something like 400 individuals, 2 million cells. So that was a really useful sort of resource for us that, in terms of data integration, we just used, I think, SCVI or ScanVI at the time. So essentially these conditional variation autoencoders that I was showing you before. So just sort of these pure conditional autoencoders. But it's useful. You can do all kind of things on top of that, right? Because now you have many subjects. You can do association of cell types with particular downstream markers, patient representation learning, things like that. And we do find interesting things because you can map disease on top of it. We find, for example, a shared disease state in monocyte-derived macrophages where this factor SVP1 is high. That's actually shared across pulmonary fibrosis, COVID-19, and also some type of carcinoma. So I think that's actually interesting. And we've been doing this now at scale together with the Human Cell Atlas for other organs, not just for the lung, but also for the pancreas, particularly for the islets at the moment. We're doing this for the whole pancreas. It's currently ongoing with Rui Chan's lab for the human retina, and most recently with Barbara Treutlein and Greg Camp's lab for brain-derived organoids. So we're sort of always doing this type of integration work. But you get then these individual dimensions, and these are going to be hard to interpret. So how can we do a hybrid model for that as well? So there's I think that's really a challenge that maybe some of us have been addressing by basically taking linear models, but often has been ignored. So you have these gene time cell matrices, and for most of the downstream analysis, such as clustering, integration, and so on, we want to have some type of latent representation. This used to be, for a very long time, this KNN single cell graph. With increasing complexity and scale of the samples, often it's been sort of replaced by a latent projection. And this latent projection could be something linear, but often is then these sort of new network or KNN integrated or whatever type of intermediates, right? So that's where we use these autoencoders, but then this thing turns non-interpretable anymore. It's kind of hard to go back to the original genes, right? So you always need to do this sort of index-wise. For individual ones you can do, but for gene programs it might be a bit more challenging. So these latent spaces are crucial, but they're difficult to interpret. And the solutions now to make them is that you either add really biological priors or just purely statistical ones. And I think it's important to say that this general question of finding a nonlinear function that gives you an optimal independent, it's often called disentangled meaning that if you sort of change this one variable in ground truth, only sort of one thing changes. This disentanglement is not possible if you don't add these inductive biases. So you need to do something like that. And there's various approaches. The purely data-driven ones could be, but maybe could you add also principal ones? So early on, for example, we sort of assumed some type of sort of decorrelation in that gene expression in the cell cycle space, which is wanted to take out known factors such as cell cycle. In a very nice perspective, and in many downstream applications has been sort of proposing just doing factor decompositions. And examples such as MOF are very popular, particularly multimodal settings, where you can sort of find the factors. So you sort of essentially find some type of independent decorrelated or something like that expression. So you do sort of fancy versions of PCA or IC or something like that. Other approaches are to add actually biological priors. Oh no, okay, no, there's some nonlinear ones. Sorry, I missed it. So there's we've been doing a workshop paper where we add some type of constraints to the VAEs. So some type of statistical constraint in the VAE, and we did a recent thing where we add actually gene program priors in the decorrelation, so you have a linear decoder. But these are all kind of, I mean, they work, they give you some latent space, but you always pay at price. So what we now asked is, can we do this disentangled representations where, find this particular dimension where, let's say, one cell type sort of is sorted out, or you sort of find a dynamic process? Can we do this sort of a bit more systematically? And here, I want to show you results from Amir, PhD student in the lab who has been sort of hitting his head against that problem for some time, but I think found a really exciting idea. And here we build upon something that's called additive decoders that has been proposed at NeurIPS just a year ago, where there's really some nice theory on actually showing that you can do disentanglement. Essentially, the idea comes from computer vision, where they have this idea of sort of having this additive scene. Sort of you put you put a chair in the picture and you sort of have this additive composition of scenes, right? But then these effects often are non-linear on the pixel space. So you have this additive scene, but you have sort of this non-additive behavior. So the latent space is additive, but the pixels, so the observed space, is something non-additive. And how can you bring these things together? In a sense, this is sort of what we want to do with gene programs, right? So this is the idea of the model. We take our typical variational autoencoder, conditional variational autoencoder that I showed you before. So you take the variables, encode them, you decode them again. But now we take this additive decoder. So we take the latent representation, but now we cut them in each of these individual dimensions. This could be univariate, it could be multivariate. We add the condition to that, and then we individually decode them. So we have a nonlinear decoder here, so a nonlinear function, but just on much smaller dimension, typically one-dimensional plus covariate. And then you get each of these representations. You do a lot of these decoders, and then you combine them with a pooling function. And here you do something nonlinear. For example, log X type of pooling. And what we can show is that these what we call pooled additive decoders, that actually then enable really general disentanglement. So this additive decoder disentanglement theory we can sort of pull up to this scale. And that's really exciting. That gives us some theoretical grounding that we can actually do this now. So we use log sum pooling for this, and I just show you now that this actually works. So in this case, we take this lung atlas that I was showing you before, and we just look at the embedding. And first, it's important to say that they're pretty much as expressive as before. So the latent space, you find the same thing, and the integration scores are pretty much the same as for SCVI. But you can now look into individual dimensions. And just on this UMAP, I plot you now a few of those dimensions where they sort of localize in this sort of cell type arranged space. And for example, here, okay, we find that here there's, for example, one particular cell type in there. This would be dimension 17, right? And if I sort of just show you a few interesting dimensions that are now really disentangled, here you find a process that sort of seems to be enriching in some sort of set of T cells and a shared process. And then you also see some type of developmental dynamics here. If you plot those two against each other, you actually see that there's some sort of erythroid development happening. How can we quantify this more consistently? Well, because now we have individual dimensions, we can actually sort of play around with these, right? And this is the idea how we really make them interpretable, we sort of traverse not just one dimension. So we change one dimension and see what the effect in the gene expression are, right? So we just correlate them with the genes, or we identify then also relevant genes. Okay, and then you sort of get the gene programs, things like that out. So this is what we now do for these various factors that I showed you before. And then you get a bunch of transcripts out that are, for example, here markers for dendritic cells, for Treg cells, for some type of stress program and so on. And if I now just superimpose these here on these pictures that I showed before, is, oh yeah, here we pick up these MDCs. Here we sort of see this Treg substate. The shared process here is actually this association that's sort of seen all over. And these two here would be actually markers for erythroid progenitors and for erythropoiesis. So you really get this latent space with pretty much similar integration quality, but now with sort of each axis meaning something. Let me try to convince you that this is actually really systematically happening. And we have some theoretical grounding, but things in practice at scale and these high dimensions could always go wrong. So what we do now here is that we take now a ground truth variables and we just look at changes in one representation. And this is what we do by trying to formulate some type of ground truth. How can we do this? Well, let's say one ground truth could be known cell types. That's what I showed you sort of a bit before. Another ground truth could be if you attach particular perturbations, you could see where they go. Or maybe you have some underlying process. And then you want this to be only present in one of these latent dimensions. That's kind of what we measure. For this, we come up with some type of scores. And this is what I visualize you here now, right? And I compare, we call it DRVI for disentangled representation, variation inference, or DocDVI is sometimes called. And there's an additive pooling version of that. We mostly use this one to sort of previous approaches, linear ones, as well as nonlinear embeddings. And what you now see is that you always look at the average gain over this most simple linear PCA model, and then you see that DRVI sort of always outperforms across a bunch of data sets, a developmental one, perturbation one, and various batch ones at scale. Won't show you details too much on this, but I just want to show you that this actually works also in the lung. Before this was just a PBMC example, sorry for that. So if we do this here now in the lung, we get basically all these various factors out. So I plot you now here the different cell types, and I plot you which dimension sort of picks up the cell types. And you see that you really get pretty much dimension resolved for those various cell types, and then you get some sort of shared programs. So you really get now these interpretable axes in. And you can look into what's this shared one here. And for example, you see this one is sort of giving you some type of dissociated stress response, one myeloid one, and so on. The latest point I want to make is that now that you have a method that sort of finds individual dimensions, then you can ask, like, how much does it depend on the scale of the latent space? Like, is there maybe even like a sort of lower dimensional space that you would want to look in this whole thing altogether? And that's actually what we observe. So if we now increase, so here just we take latent space size 32, we see that the size of the component at some point drops just at the end. But if we go to a larger one, it seems to be dropping again a bit later. So it needed that capacity, but drops. And if you go to 128, sort of it takes a little bit more, but it starts dropping again. And this is for the mu data set. But we kind of think there might be maybe some sort of maximal intrinsic data dimension or something like that in there. And this is actually what we see if we do this across those data sets that we benchmarked before. We see that there seems to be sort of going up, but at some point then thresholding the number of dimensions that's being used. And, this is starkly different from PCA or ICA or so, where you always sort of add additional sort of low variance components you keep adding. Here sort of really starts dropping. Big exciting phenomenon, and we're currently studying this. So to summarize this, I think in order to build interpretable latent space, potentially also atlases, we build them by integration of signature profiles. And this we do with conditional rational autoencoders, but with DRVI, we can do so in a more interpretable fashion. So if we now have the atlas done, what do we do with these, right? So using those latent spaces, I think, becomes very important. And I think for this sort of like transfer learning, I think is the way to go, where you sort of have now your given atlas and you superimpose, you map sort of cell states on top of this, right? And we've been coming up with different ways how to do this. I think maybe quite exciting one is, and I just wanted to briefly advertise this here and mention, if you now have patient like sample level covariates, this could be like a disease classifier. This could be actually also something like QTLs or so. Like, this could be something like SNPs or so, where you can then call QTLs, which, typically are shared across all these cells. What you can do is you can do the aggregation across the individual cells using an attention layer. And we've been sort of setting this up here. We encourage you to check this out, or be happy to chat about it. What I just want to wrap my talk with, though, is this extension towards these sort of really large scale models at the end. I was mentioning generative AI, and of course, the big kid on the block at the moment is this extension towards these large scale models called foundation models. And I just want to sort of put this a bit in context, because I think this idea of acting on these cellular landscapes is a really old one. In systems biology, this has been asked 20 years ago by these things, what they at the time called whole cell models. I'm sure you've seen at some point. They sort of, with the Human Genome Project being done, the hope was now we have all the bits and pieces, we can sort of put this together, put structure and everything, and then we build this very big thing. But then the predictions looked really poor, and just doing this in a purely first principle version kind of didn't work. And even though this has been sort of put forward, I think in principle these things were not actionable, and it's kind of failed. But there's renewed interest. We have these data sets. We have these different modalities that can measure at the same time. So, Dimas Hasabe some time ago also mentioned, he wants to do a virtual cell in the next 10 years as one of his big dreams. CZI has been putting forward, particularly Mark Zuckerberg and Priscilla Chan have been putting forward to build this virtual cell modeling system as a thing to sort of lead to new breakthroughs. So I think this idea, putting these things together, is becoming really exciting again. And here again, we can use, in principle, this is, given now these latent spaces that we do more and more across atlases, I think it's actually a good setting, right? So what you would do is you sort of do some training on these large scale samples, maybe masking of particular genes so that you find this embedding, or you take the evolutionary autoencoder ones that we showed before, and then you do fine tuning for these downstream tasks. So this is sort of the key idea. Here we just put out a review of existing methods. I think the seminal one that really people that really got excited about it from Christina here, where you take single-cell RNA-seq, you learn in a self-supervised fashion a transform, and then you downstream fine-tune it for cell-level tasks such as cell annotation in particular, but then also for gene function, things like that. I think at the moment the vote is still out how well this actually generalizes and is sort of really outperforming simpler, linear, potentially more constrained models. But I think that the vision of such sort of train them once across very large scales, I think is very exciting. We have a model where we actually add spatial constraint as an additional sort of harder fine-tuning task, where we think these things such as cell clustering, cell annotation is just too simple a task where you won't outperform linear models in many cases. And then actually we see that if we do this really at scale, and this case at scale really means for us training across more than 100 million cells, which was really hard from all kind of engineering perspectives, so we don't even go into. We can do such tasks such as niche prediction, where you give us a gene expression profile just of a single cell, and you predict its local niche composition. And this is actually, of course, in a fine-tuned version for various organs. Very excited about this direction. Just wanted to flash this here, and I'm really looking forward to seeing how these new directions develop. Why? Well, we said we want to map things on top, and this is sort of mapping on top, like building like an actionable atlas, so to speak. So once you have these models, I think enabling then this lab in the loop, where you hypothesize like some new things, you test them, and you sort of go back. I think this is going to be really exciting. So these agents that then sort of act, who will maybe sort of support us in our research. Yeah, that's maybe one of the visions to go. So with this, and we've been playing around with this, and I think we have a really exciting solution, not just for bulk averages, but for sort of phenotype prediction that I encourage you to check out. Really happy to chat about it, but this is a bit off topic, so I've been not talking too much about that yet. We call it PROPHET, the predictor phenotypes. So with this, let me conclude. With generative AI, we can learn cell atlases and we predict perturbation effects. And I showed you two new approaches that we've been doing. One is called RigVelo, where we do dynamic modeling with gene-related network constraints. And then we can really do this sort of dynamic perturbation inference. And a DRVI, where we do this disentanglement of the latent spaces, thereby being able to interpret them. Both are not yet published, but we hope to bring them out soon. Challenges, as it was mentioned before, is this virtual cell. Of course, this could be sort of digital twin, like how much constraints do we actually want to build in? I think those old purely first principle-based ones kind of didn't work, so we need to have them data-based, but maybe there's a room for these additional constraints. All of this needs more benchmarks. And in the end, this will get really large. So how do we work on more than 1 billion cells? I think is a big challenge. And I think I was mentioning already this lab in the loop as one of the sort of key applications where you would go. I think I acknowledged already all the contributors and partners. So I just want to finish by showing my lab. I'm really acknowledging this fantastic team, grateful for funding partners, and I thank you very much for your attention.
All right, thank you very much. So we have some questions on the chat room. I'll read off it for you. The first question is, in RegVelo, have you tried using transformer instead of autoencoder? And if you think autoencoder more fits in the algorithm, what is the reason? Please.
I think at the moment it's basically not yet shown that a transformer-based representation really gives us a more robust first representation, but in particular then also being able to reconstruct cells. With a transformer, you can sort of get very robust latent spaces, but the sort of decoding is kind of hard. So for that, we still use rational autoencoders. And in fact, actually also in computer vision, where that's sort of the example, in addition to language, where transformers beautifully work. Vision transformers also always have a encoding decoding stage from the actual full pictures to the sort of large, smaller ones where you apply transformers. But it's a good point. We actually have a model that's still sort of very much in work in the lab, where we combine this also with the transformer-based representation. Not the dynamical part yet, but the perturbation prediction.
Okay, so the second question is, do you think it will free us from using research approach to find optimal and latent when using NCVI tools?
Yeah. I mean, it's kind of cool, right, that you find this intrinsic one dimension in the data. I do think it has quite a bit to do with how much sort of initial noise you might have in the data. So I think selection of highly variable genes, or however you process before, could still play a big role. But in a sense, yes, the hope is that if you just take sufficiently high dimension, and not the hope, I mean, that's kind of what we've shown. We've shown this really systematically across a whole bunch of data sets, that in the end it sort of finds a consistently smaller set of useful components, and the rest is going to be set to zero. So yes, you don't need to select low ones. Sometimes you have reasons that you really want to compress your data in a higher fashion and sort of more compressed fashion, then you can throw out additional ones. But yes, so there seems to be an upper threshold that seems to be also correlating a little bit with the complexity of the data set. So we have one example where we look, I think, in a zebrafish embedding, like a whole organism one, and then you actually need a higher number of components than maybe in this simpler PBMC settings or so.
Okay, thank you very much. So the third question is, since RegVelo utilize both GRN and RNA velocity, from GRN perspective, we can get like benefit from multiome data, but for RNA velocity, since multiome data is created by nucleus, do you think unspliced or spliced transcripts efficiently reflect the velocity?
Yeah, very important point. I think there are a few papers, we've been also trying to show this a bit, that you can actually call some set of dynamics also from purely single nuke data. But of course, you have splicing machine is sort of less expressed there. And the big sort of the high level answer I have to this is that the model at the moment, we've been focusing on purely splicing. But there's sort of various versions that you can add to constrain also this to get additional temporal information. So one thing you can do is that you add, and this is what we've been playing around a bit with, is metabolic labeling, right? And this, of course, you can then sort of read out with any of the two assays, and that allows you to then connect time points. This is what Dynamo was doing. And because we have this very flexible full dynamic model, you can add this also as a measurement observation. You can actually completely drop also unspliced reads, and you just sort of formulate the whole model only on full RNA. And then you can combine this with additional information such as time points, which is what we sort of showed as an example in 7.2. The moment we focus on the pure velocity example, which seems to be really doing a lot of good things for us at the moment, but mostly good for cytoplasmic one.
Okay. And please feel free to ask any questions from both, like from the floor and on the chat room. All right, so if you don't have any questions, we will close the session. All right, thank you very much, Fabian, and please give a big hand to Fabian for the great talk. Thank you very much.
We hope you find new ideas and inspiration at the 2024 SNU-GMI International Symposium. It's been an honor having such a talented and passionate group of professionals with us today. Please note that today's recording will be available on the Genomic Medicine Institute YouTube channel, so you can visit anytime to review the sessions. We look forward to seeing all of you next year and continuing our journey together in advancing genomic medicine. Thank you.